Good morning, Professor, and to fellow classmates. Today, I will be presenting you a global issue that has plagued both healthcare and public settings throughout time. This problem is known as Mrsa. What is Mrsa? Mrsa stands for methicillin-resistant Staphylococcus aureus. It originated from the species Staphylococcus aureus that evolved and mutated to be resistant to multiple drugs and antibiotics. Mrsa is a single-celled organism from the domain bacteria. The name coccus, as you can see in Staphylococcus above on genus, refers to the shape of the organism, which indicates that it is conical or spherical in nature. There are various strains of this organism. However, I will be focusing on this particular species that throughout time has developed multiple drug resistance to beta-lactam antibiotics, known as Mrsa. Mrsa is GPC. What does GPC mean? It stands for gram-positive cocci. Gram-positive is a result that is derived from staining procedure called the gram staining test. The purpose of this test is to determine the chemical makeup of the cell wall on the bacteria. The gram stain test classifies the organism into two broad categories depending on the cell wall. When the cell wall stains positive, it will appear purple, which will denote that organism has a thick peptidoglycan cell wall. Contrastingly, if the organism has a thin peptidoglycan cell wall, it will appear red during the staining process. As you can see on the slide to the right, Mrsa is gram-positive cocci, which concludes that it has a thick peptidoglycan cell wall with the organism's shape being conical or spherical. So, unlike other strains of Staph aureus, Mrsa is genetically distinct in that it has the ability to survive through multiple antibiotic therapies. This is done through the acquisition of extrachromosomal elements containing genes that confer resistance to certain antibiotics. It is initiated through methods such as horizontal gene transfer through sex pili, plasmids, and transfer genomic islands, and then replicated through vertical transmissions to future generations. For Mrsa, there are two important components that make this mechanism work. SCC-MEC, which stands for staphylococcal cassette chromosome, is a genomic island that contains additional genes associated with antibiotic-resistant gene MEC-A, and there is MEC-A, which is a biomarker gene responsible for resistance to beta-lactam antibiotics. To simply put, the combination of these two important components allows MEC-A to encode penicillin-binding proteins, resulting in peptidoglycan cross-linking and enabling cell wall synthesis even in the presence of antibiotics. Therefore, this has become an issue to healthcare professionals when treating Mrsa. This issue has plagued the global community and has become an enigma for both scientists, researchers, and doctors and healthcare professionals worldwide. And Mrsa has become even more prevalent in healthcare settings. Proper diagnosis from a healthcare professional is required in order to differentiate whether the organism is Mrsa or not. In humans, Staph aureus usually resides as part of normal microbiota in the skin, the respiratory tract, and gut mucosa. It actually has a symbiotic relationship with us. However, when things take a turn for the worse, it appears as red bumps on the skin, sometimes resembling pimples, spider bites, and even boils. This can be accompanied by fever and other skin problems like rashes. The problem can be elevated within a few days, where the bumps become more larger and painful, where they can open into deep pus-filled boils. Modern data suggests that 75% of these infections are localized to the skin, but usually they can be treated. Because Mrsa can be found in and on many parts of the human body, it has many modes of transmission. Transmission routes may differ depending on whichever setting it is in, but usually it enters through a cut or an opening on the body. The setting can be a hospital-acquired setting or a community-acquired setting. In hospital-acquired settings, the risk factors are elevated because the patients are more vulnerable. This can be due to age, or if the patient is immune compromised, and or adverse side effects of other antibiotics that the patient might be taking. Other risk factors may include invasive insertable medical devices such as catheters, prostheses, and drains. Last but not least, the aggregation of sick people in confined spaces make the disease more transmissible, and this is also known as nosocomial infections. In community-acquired infections, this is usually done through contact sports such as boxing, basketball, and in living and other types of sports like that. This can also be transmitted by living in crowded or unsanitary conditions, using recreational intravenous drugs, and engaging in sexual risky behaviors. So what can we do to prevent this infection from occurring? For community-acquired infections, we can start by washing our hands. Our hands is the first thing that usually makes contact with anything. Carefully washing our hands is the best defense against germs, and this also applies to other germs as recent as COVID-19, where the media has continuously endorsed hand washing to curb off infection rates. Second would be to keep topical wounds covered, such as abrasions or cuts that need to be first sterilized and bandaged until they heal. Pus from infected sores may contain Mrsa and co-infect healing wounds if they are properly covered. Personal items such as razors, toothbrush, towels shouldn't be shared. These items can harbor Mrsa and can be transferred from person to person. Staying hygienic by washing towels, linens, and athletic equipment can cut down on Mrsa transmission by killing off the organism being harbored on these items. Last but not least, non-use of recreational drugs through intravenous means will also cut down transmission. In hospital-acquired settings, the transmission rates are usually higher. Therefore, prevention methods are usually more strict, and contact precaution measures should be more methodical. Anyone visiting patients should be properly screened and should be given the proper PPE. Hand washing is also an important component for both healthcare professionals and patients alike, as to mitigate infection. For patients that are immune compromised and those with active infections, isolation or quarantine can help curb the spread of Mrsa. Due to adverse side effects, certain antibiotics are restricted from use. Glycopeptides, cephalosporins, and quinolones have been associated with risks of colonization of Mrsa. In a clinical setting, there are various methods of identifying Mrsa. Diagnostic testing through reference labs are usually key methods in identifying the organism. In microbiology, specimen samples are collected usually through blood, sputum, and bodily fluids. These fluids are then inoculated using aseptic techniques on a culture media, either selective or general, for enumeration and or isolation of a certain organism. This media is then incubated between 24 to 72 hours, where the cultures will grow for that duration of time. In the case of Mrsa, traditionally blood mannitol salt agar or Baird Parker agar containing methicillin or oxacillin has been used to identify and sensitivity of the organism. Showing resistance or sensitivity to the selective media helps the organism identification through process of elimination. Other methods of testings are done in conjunction to verify the existence of Mrsa. This includes microscopic examination and morphological testing of the media to verify Mrsa. As you can see in the picture above to the right, zone of inhibition shows resistance to oxacillin disc on the Mueller-Hinton agar. However, this method of testing takes critical time that could be used to treat the patient. Therefore, there are alternative methods of testing such as rapid testing by using quantitative PCR. Polymerase chain reaction tests detect Mrsa through nucleic acid amplification through certain markers. Rapid agglutination tests look for certain markers, or certain proteins in this case, that binds to penicillin, which Mrsa uses to be resistant to oxacillin. This will give a positive identification that the organism that we are looking for is Mrsa. Because Mrsa is known to be resistant to many types of drugs, the drug of choice to treat Mrsa tends to be vancomycin. Routes of administration for antibiotics can be varied, but usually are given via IV, oral, and/or a combination of both depending on the circumstances of the patient. For topical infections, treatment options include removing infected tissue, incision and drainage of the infection, and application of antibiotics to the infected site. Internal localized infections such as respiratory infections require hospitalization and intensive care to combat the disease. This concludes my findings regarding Mrsa. For reference, I have posted the links to the websites that were used in research to the Mrsa topic. Thank you and have a good day.